The overall goal of this procedure is to detect low-frequency somatic mutations in a variety of sample types.
This methodology can help answer key questions in somatic mutation testing by facilitating the detection of very low-frequency mutations. Here we'll look for mutations in the MYD88 gene in bone marrow samples. The main advantage of this technique is that it provides highly accurate and sensitive information about the presence of somatic mutations, even with very few neoplastic cells.
This wild-type blocking PCR-based sequencing assay was developed to amplify part of exon 5 in the MYD88 gene, ensuring coverage of the L265 hotspot. The forward and reverse primers were designed with a 5' M13 sequence to allow for annealing of complementary sequencing primers. Design the blocking oligonucleotide to be approximately 10 to 15 bases in length and complementary to the wild-type template where mutant enrichment is desired.
A shorter oligo will improve mismatch discrimination. To achieve high target specificity, it's important not to use too much of the blocking nucleotides, as this will result in a very sticky oligonucleotide.
To design the blocking oligonucleotide, begin by navigating to the OligoTools website. Select the OligoTM prediction tool. A new window will open up. Paste the sequence of the wild-type template to be blocked into the oligo sequence box. Add a plus sign in front of the blocker bases to mark them. Click on the Calculate button to determine the approximate TM of the DNA blocker hybrid. The calculated melting temperatures will appear in